Hello, my dear classmates and viewers. So, from the previous discussions that we have tackled, we already identified the components of the cell structure. This time, we will dig deeper, particularly the plasma membrane part of the cell, and elaborate its structure and function. Now, for the first topic, we will discover more about the border of the cell and its structure, the plasma membrane, or in other terms, the cell membrane. So, a brief overview of the plasma membrane. Plasma membrane is a specialized structure that surrounds the cell and its internal environment. It also controls the movement of substances into or out of the cell. Therefore, the plasma membrane can protect the cell from the outside environment, and it can determine what can enter or leave the cell. This property is known as semi-permeability. Something can enter while other things cannot. Because of this property, this semi-permeable membrane allows certain substances to pass through it. So here, we'll take a closer look at the different components of the plasma membrane, examining their roles, their diversity, and how they work together to make a flexible, sensitive, and secure boundary around the cell. The currently accepted model for the structure of the plasma membrane, called the fluid mosaic model, was first proposed in 1972. According to the fluid mosaic model, the plasma membrane is a mosaic of components, primarily phospholipids, cholesterol, and proteins, that move freely and fluidly in the plane of the membrane. In other words, a diagram of the membrane, like the one below, is just a snapshot of a dynamic process in which phospholipids and proteins are continually sliding past one another. So here, the principal components of the plasma membrane are the lipids, which are the phospholipids and cholesterol, proteins and carbohydrate groups that are attached to some of the lipids and proteins. So let's explore these components one by one. So first is the phospholipid. Well, this is the main building block of the plasma membrane. So phospholipid is a lipid made of glycerol, two fatty acid tails, and a phosphate link head group. Biological membranes usually involve two layers of phospholipids with their tails pointing inward, an arrangement called a phospholipid bilayer. So let's delve first with the phospholipid. The structure of the phospholipid contains the phosphate head. The second is the glycerol backbone and the two fatty acid tails. The hydrophilic head contains the polar phosphate group and the glycerol backbone. Since the head is polar, it is hydrophilic or water-loving. On the other hand, hydrophobic tails contain the saturated or the straight carbon chains and the unsaturated or bent carbon chains, also known as the two fatty acid tails. Since the tails are nonpolar, this part is the hydrophobic region. So the way this picture depicts, we give the phosphate head group kind of like a head and the two fatty acid tails hanging down from it kind of like strings on a balloon. The glycerol backbone is actually what it sounds like. It is what holds the fatty acid tails to our phosphate head. So this structure actually has a very interesting property. Up here, this is the head group. It is actually hydrophilic or polar. So hydrophilic means that it's water-loving. This phosphate head group will do whatever it can get to water. It loves water. But these fatty acid tails, because they are very, very long carbon chains, this is hydrophobic. So it's water fearing. these two fatty acids will do whatever it can get away from the water. So here in this picture, we have the plasma membrane cross-section. Notice that the plasma membrane arranges or behaves like this. So just why? As of what I've said about the previous slide, the phospholipid molecule has both the hydrophilic and the hydrophobic parts together. Because the molecule has these two rings, it is called the amphipathic molecule. So in water, what would this do is that the hydrophilic heads wants to be close to the water as possible, but the tails don't. What will happen is this: the phosphate groups are going to cluster together while the tails try to shield themselves away from the water. Now, the inside of the cell too has also the water substance in the cell called the cytoplasm. So it kind of arranges the phospholipid molecule in a bilayer form. So this is actually forms a really unique structure because the fatty acid tails are going to start grouping like this here, here. And the phospholipids are going to be kind of upside down so the phosphate head groups can be close to water while these tails can be hydrophobic and get away from the water. Hence, we arrive on the term phospholipid bilayer.
Membrane fluidity. Uh, the structure of the fatty acid tails of the philosophids is important in determining the properties of the membrane and in particular how fluid it is. Also, membrane fluidity is affected by fatty acids, more specifically whether the fatty acids are saturated or unsaturated has an effect on membrane fluidity. Then, the saturated and unsaturated fatty acid tails of philosophids behave differently as temperature drops. At cooler temperature, saturated fatty acids can pack tightly together, making a dense and fairly rigid membrane. It is because saturated fatty acids have no double bonds in the hydrocarbon chain, and the maximum amount of hydrogen, the absence of double bonds, decreases fluidity, making the membrane very strong and stuck lightly. Then, a membrane containing unsaturated fatty acid tails will stay fluid at lower temperature than a membrane made of saturated ones. It is because unsaturated fatty acids have at least one double bond, creating a kink in the chain. Take note, the double bond increases fluidity. Then next is the structure of phospholipids. If the phospholipids have small tails, they may have formed a micelle, a small single-layered sphere, while if they have bulkier tails, they may form a liposome, a hollow droplet of bilayer membrane. A liposome is a closed spherical lipid bilayer carrying aqueous solution. A micelle are lipid molecules that arrange themselves in a spherical form in aqueous solution. The formation of micelle is response to emphatic nature of fatty acids, meaning they contain both hydrophilic region or polar head group as well as hydrophobic region or the long hydrophobic chain. Cholesterol. In addition to philosophids, animals have an additional membrane component that helps to maintain fluidity. Cholesterol is another type of lipids that is embedded among the philosophids of the membrane. It helps to minimize the effect of temperature on fluidity.
This time, we have arrived at the second main component of the plasma membrane. These are the variety of proteins, also known as the membrane proteins. So a membrane protein is a protein molecule that is attached or associated with the membrane of a cell or an organelle. The functions of the membrane proteins are the following. First is a transport, which aid movement of chemicals into or around the cell. Second is enzymatic activity, acts an enzyme built into the membrane with active site exposed. Third is the signal transduction, built into the membrane with active site exposed. Fourth is the cell-to-cell recognition. Some glycoproteins serve as identification tags that are specifically recognized by other cells. And intercellular joinings, membrane proteins of adjacent cells hook together to form various junctions. Membrane proteins can be put into two groups based on how the protein is associated with the membrane: the integral membrane proteins and the peripheral membrane proteins, and of course, the lipid-anchored proteins. So first, integral membrane proteins, also called intrinsic proteins, meaning integrated into the membrane or permanent fixture on the membrane. Note that they have at least one hydrophobic region that anchors them to the hydrophobic core of the phospholipid bilayer. They function as transporters, channel linkers, receptors, proteins involved in accumulation energy, and responsible for cell adhesion. Integral membrane proteins also can be classified according to their relationship with the bilayer. Transmembrane proteins, which extend all the way across the membrane, and the integral monotopic proteins, which stick only partway into the membrane. So the transmembrane proteins, also called the integral polytopic proteins, span the entire plasma membrane. Their function is mainly to regulate the transport of specific molecules across the membrane. There are two basic types of transmembrane proteins, the alpha helical and the beta barrels. Depending on the number of transmembrane segments, transmembrane proteins can be classified as single-span or bitopic and multi-span or polytopic. Now, I would like to emphasize this. This protein corresponds as bitopic because they span the lipid bilayer only one time, here and here. The polytopic, on the other hand, crosses the membrane multiple times, thus called multi-span. So next are the integral monotopic proteins. They are permanently attached to the lipid membrane from only one side and do not span across the membrane. So here is the overview of the proteins, which are identified based on how often they span the lipid bilayer. So monotopic here it is, the bitopic and the polytopic.
Here are some examples of integral membrane protein. Number one is the Na plus or K plus ATPase, responsible for establishing and maintaining the electrochemical gradients of sodium and potassium ion across the plasma membrane. Number two, glucose permease, the reversible transporter protein of glucose. Number three, ion channel and gates, the flow of ion across the cell membrane. Number four, gap junction protein, a direct connection between the cytoplasm of two cells which allows various molecules and ions to pass freely between cells. And lastly, the glycoprotein. It is the cell-to-cell interaction. Then
The next class of the membrane proteins are the peripheral membrane proteins, also called extrinsic proteins. They are temporarily attached to the lipid bilayer or to other integral proteins. Most are hydrophilic, so usually they are either attached to integral membrane proteins or they can directly bound to a polar head group of the bilayer. This way, they can be easily removed, which allows them to be involved in cell signaling. They are often associated with ion channels and transmembrane receptors.
And here are some examples of peripheral membrane protein. Number one is spectrin. Number two is kinase C. Number three is phospholipases. And number four is hormone receptors. Then lipid anchored protein are protein located on the surface of the cell membrane that are covalently attached to the lipid embedded within the cell membrane. Here are the overall, there are three main types of lipid-anchored proteins, which includes prenylated protein, number two is fatty acylated protein, and three, the GPI. So, perenylated protein are protein with covalently attached hydrophobic isoprene polymers and cysteine residue of the protein. The fatty acylated protein are protein that have been have been transitionally modified to include the covalent attachment of fatty acids at certain amino acid residue. And the GPI proteins, it plays an important role in embryogenesis, development, neurogenesis, and the immune system and fertilization.
So this is the summary of the membrane proteins based on how they are associated with the membrane. So integral to membrane, integral membrane proteins only. Intrinsic to membrane, integral membrane proteins together with lipid-anchored proteins. And finally, extrinsic to the membrane are the peripheral membrane proteins only.
And lastly, carbohydrates. Attached to protein or lipids on the extracellular side of the membrane, forming glycoprotein and glycolipids. The plasma membrane structural components. Figure 1. Overview of the plasma membrane structure. Parts of plasma membrane. Its components. Pilosophids. Its location is in the main fabric of the membrane. Number 2 is cholesterol, tucked between the hydrophobic tails of the membrane pilosophids. Number 3, integral protein, embedded in the philosophid bilayer, may or may not extend through both layers. Peripheral proteins, on the inner or outer surface of the philosophid bilayer, but not embedded in its hydrophobic core.
So this is the end of the first topic out of the four topics of this chapter. Thank you, and let's explore deeper by on the next topic, which is the passive transport role of the plasma membrane.